science students, or I should say not science students, more specifically biology students. Uh, some of you know me, but just a quick introduction. My name is Larry Stewart, and whether you are a Nevada High School student, whether you were a Crowder bio student, uh, welcome to our first of, unfortunately, many video lectures as we either talked in class or will talk in class based on which school you are. And with Crowder, you probably are seeing this before seeing me. At least that was the intention. And with the high school, I'm sure we have come together for those few introductory days, and you're possibly watching this over that first weekend. But either way, hey, welcome. I look forward to this journey. Just a little bit about myself. Not that that's important. I mean, who really wants to know about an old person, what they do? But anyway, um, again, Larry Stewart, science teacher for a tremendously long time. Um, Way too long to even think about. But just looking forward to this opportunity. This video lecture series is part of uh, the processes that have happened the last couple of years. Um, if you're a Crowder student and this is, you've been at this a couple of years, maybe you were around in the summer of 2019 when the computer virus kind of took over the Crowder system and kind of shut us down and affected it all the way until the fall semester. But we're all certainly aware of what is happening currently and what has happened since early part of March, and that's the COVID virus. So this whole process is going to be kind of set in place. Both campuses, the high school and Crowder, and of course, school settings all over the country are looking for ways to best kind of utilize what's going on and that combination of in class, online, the whole kind of gamut. So what I'm going to do is, and if you've been in a high school setting, we've already talked about this. And if you're at Crowder, we will talk about this come our first class session on Monday, the 17th. But anyway, this process is going to be, it's called a flipped classroom strategy. In other words, it flips the time usage. Instead of giving you homework to do when you're away from the classroom and then lecture in the classroom, we're going to flip that to where you will watch the video lectures, taking the notes. And then when we come to class, we will spend that time just learning and reinforcing. In other words, kind of doing the homework in the room and the lectures outside the room. That's the flipped terminology. And what that will allow us to do is if something does happen and we do have to be away for a period of time, we can have a situation in place where you're already accustomed to the getting information in a different format away from me, and then I can just follow that up with the material needed to learn the material as well. So it's just a way, if nothing happens and we go through an entire semester with no changes, great. But if there's one thing that we've seen in the last couple of years with school settings is that we can no longer assume day-to-day as is operations and therefore have to have a more flexible situation. That's what this is all about. So, but again, welcome. I appreciate you watching this. Again, the expectations will be that you take a full, complete, quality sets of notes. And in both school settings, we'll have different ways to monitor that. At the Crowder level, It's simply going to be, hey, you had the opportunity. If you chose to, fantastic. That will increase your ability to efficiently learn the material. At the high school setting, it will be a part of your homework. And I'm using air quotes there, which you cannot see. Point system, but again, we've already talked about that, so you know how that is going to work. So the purpose of this is to kind of begin that session. So chapter 1, as you've seen in your syllabuses by now, or syllabi, I guess is the proper way of saying that, to sound more collegial, is to kind of begin that process with chapter 1. Chapter 1 is a unique chapter. Its purpose is to kind of just set up the rest of the book. And so most of it is either just background information or side information that is interesting, but not necessarily something to focus on in a testable manner. But the one part of chapter 1 that we are going to focus on, as you can see there by what's on your screen, is the method in which scientists use to determine results, or to learn about things, or to find things out, to determine what ways things actually worked. When I was your age, or in your position, if you're a student who's older, maybe, I shouldn't have said age, but in your situation, especially at the high school, middle school, that kind of stuff, it was called the scientific method. I've got the SM on there just simply because that's the way I've referred to it for hundreds and hundreds of years. But the scientific method, I meant also to add about doing these video lectures. That way the lectures can be conducted in a way in which I'm not obstructed with mask and worried about spacing and moving around and communicating with you in ways, and this also ways you can you can take the notes and go through the lecture series kind of in the comfort of your own space, whether that's at home or work or on the couch or just wherever, in your own room. You can listen to it in a manner that is easiest for you, most efficient for you, whether it's all in one setting, broken up in smaller chunks. Watch it till you get bored, come back to it, watch some more, come back to it, whatever works best for you. Maybe what works best for you is Saturday night, 10 o'clock, Friday morning, just whatever works best for you, just as long as you're keeping up and staying with us, and just makes it easier on everybody. So with that, we will begin. Hopefully, I can keep the discussion about the scientific method down to an hour, but either if you've been with me before, or you'll quickly soon learn that I'm not always great at keeping my time restraints down. One, it's just an enthusiasm over science. It's not that it's an overcumbrance of information. I just love talking about science concepts, and hopefully that comes across as a benefit to you. If I'm excited by it, maybe not that you'll get excited, but at least you'll see the importance, and that there's actually purpose in the information. So let's talk about the method. I make an emphasis on saying it differently because these days it's not called the scientific method, and most textbooks will even talk about there's not one way to reach conclusions, and that's true. But there are general guidelines that occur, and we'll emphasize this process, as you can see kind of on the right-hand side there, just a glimpse of it. We'll use a journal, and when we come together in the classroom, we'll have this one, plus this one will also be, sure you've seen it or you saw it on the wherever we're getting this information from, whether it's Blackboard for Crowder or our Google Classroom at the high school. So you have this at your um, resource to use as I'm going through this. Yours won't be highlighted and marked up like the one you're going to see. And what you're going to see here during this lecture isn't the entire thing that you'd have if you printed it out or if you're scrolling through it. I just did screenshots of the parts that I wanted to talk about and then brought them into this whiteboard format so we could talk about how this method, this process, and I keep always wanting to say something method, but I catch myself, how this method is used in an actual scientific process with in a journal article. And when we come together, we'll have this one and we'll have some others that will evaluate just to kind of get a sense of, what these parts look like, what they are, how they're used. And so you can kind of see them in a big picture setting as opposed to me just giving you the definitions and some guidelines and an example or two. You can really see how this works. And hopefully by doing it in this manner, both here and in the classroom, it'll really drive home the importance of what's going on. And for some of you, you will find yourself in a situation at some point in your scholastic career that you will be producing or you will produce, a journal article or at least a paper over an experiment. I know for me, getting my master's in education, I had to produce a paper, and if you just have some interest in seeing that, it's in a book form now, just to make it fancy. But anyway, when I was looking to get my master's, my mentor, Dr. Janet Green, we visited with the Springfield Public School Systems and talked about or asked their curriculum director what are some things that she would like to know about different aspects of teachers or just whatever. And she threw out 4 or 5 scenarios that she would like to know more about from a data standpoint. And we took one of those and ran with it. So it is a very common situation. And if you are in the science field of some sort, especially if it's in the biology or healthcare, you could come to a point to where you also have to produce something like this. So that's enough about all that stuff. Well, lots of background information there that may or may not have been needed, but all right. But we will begin now. So here goes the method in which scientists use to determine results. All right, it's a pretty simple process. You can see by this flow chart here, and let me get a pen out here so I can. Do I get a line here? Yeah. It all starts up here at the top, and you can see this list here is a list of all of these things. But this is the flow chart scenario. So it just all starts with an observation. Whatever the field is, whatever the situation might be, it is just an observation that occurs, and usually the observation leads into a question. So, a middle school textbook would say something about the 5 senses, but we're all adults here. We understand that everything that we recognize is through one of the 5 senses, but it's just an observation that occurs, and we'll see what the observation was in this journal article once we get into it. And from that observation comes a question. And now we are right here. A question comes out of that. Whatever you were studying, whatever it might be. Okay, and then from that point, a hypothesis is formed. Okay, and we'll go over the formation of a hypothesis. But if you're a Nevada high school student or you are a recent high school graduate, at some point in time, chemistry class, biology class, somewhere, You have probably had to form a hypothesis, so you probably are aware of the structure and the importance of everything, but we will talk about that. And then from that hypothesis, now it leads into your experiment. And the experiment is designed and set up in such a way that it's to test the hypothesis. And the beauty of this article that we're going to look at is that it's a pretty complex, in-depth, thoroughly planned out, long length of time that you're going to see. But it's a pretty in-depth experiment and process. And they're not just looking at one variable. They're looking at multiple variables kind of all at the same time. And so it's pretty phenomenal. I love reading it. I mean, I don't know how many years I've been using this, but love working with it. And it's just a really good article to look at. But you'll see all that. And then it comes the analyze portion. And the beauty of what we're going to look at when they analyze the results, the emphasis that I'm going to put on is that the human element, the human factor, the human thought process is completely removed. And that is so important in a experiment process like this, that you completely take out the human interest and completely rely on the math to tell you yes, no, right, wrong, whatever. Okay, and then this portion right here, the hypothesis correct, incorrect, and if it's correct, then you go on to report your results and everything. If it isn't correct, hey, it's not that it's wrong. Okay, hypothesis is not right or wrong. Just like observations are not right or wrong. When we get into the classroom setting and we conduct labs, whether it's a high school or at Crowder, the one thing I hear all the time is, I'll be walking around looking at papers and results, and someone will have an answer and they'll say, hey, is that right? It's not important. Okay, what's important, did you record what you observed? And that's all that matters. That's all we're after. If what you recorded that you observed, if that's an honest evaluation of what you observed, then the answer is right. Now, if it isn't what I expected you to observe, then we can go back and look at the structure of what you did and how you did it and look at some of those variables. But a hypothesis, it isn't wrong or right to be correct or incorrect. It's just simply, if it's incorrect, okay, we go back, we reform a hypothesis based on what we did see, and then test again. And we just keep doing that process until our hypothesis is confirmed by the data. And when that happens, you now truly understand whatever you were studying, how it works. And that's the purpose of this whole process. Not to be right or wrong, but to actually learn what's going on. All right. So let's begin this process. And I'm going to close up my pen tray here. And we're going to slide over here, moving this over. Hopefully that didn't cause any motion sickness or anything. And as I mentioned, let me go a little bit smaller here. No, that's the wrong way. Oh, no wonder I need to go smaller. I need to be like at 70. Oh no, that's two different ones. All right, so there will work here. And I know you're thinking that, hey, you got some stuff hanging off of that, but we'll slide over and then focus on these 6 spots here in a second. All right, so as we mentioned earlier, the observation, okay, it's just a natural thing that you would observe. So let's set up the situation here. We've got these 2 guys. We got Raymond and Richard over here. Now, since I've got the highlighting all done, I can just work with my red pin here. But we got Raymond and Richard up here. Raymond and Richard, you can see they work for the Institute of Ecosystem Studies, and this is in New York. And this is essentially a company that looks at ecosystems, and when a company or some organization needs information, this is the company that you could come to either find out or get that information. But the whole background of this scenario is deer populations, and not just deer populations, but understanding the number of deer in a deer population. Here in Missouri, deer are quite prevalent. Our whole process of managing our deer population is done numerically, and part of that numbers is what they use to help set the hunting levels. In other words, the number of tags that they can issue, and that's part of the whole control and management of the numbers. I mean, you've got to, it's not just total number of deer. That's not a number that you can necessarily focus on. What you've got to understand is how many does, how many bucks, because that's also important, because you may have, and I'm just going to use the number 1,000. We have a lot more than 1,000 deer. But if I have 1,000 deer, I may say, all right, that's a great number. But of that 1,000 deer, if I've only got 200 does and 800 bucks, then we're in trouble. Okay, it doesn't seem like it today, but if each of those does, okay, only give one or two, and not all of the does will be able to reproduce. Maybe some will just die within the year. Others won't. They're not of age yet to mate. So whatever the situation may be. So it's not just an overall number, as you will see in this article, but it's numbers within the numbers that you have to also truly understand. But, and I've got it highlighted here, so we're going to start with, but the whole observation, and we will get to it in a later spot. They directly say what their observation was. But as we look at this article, the observation that they had, that there are many counting methods used by scientists, biologists, naturalists, whatever, companies, whatever you want to title them as. But there are many, 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 and they list those here eventually, counting methods used. And what they were looking to do is to devise a counting method that would be the most efficient in time, resources, and money. That way, they can have a program that when someone comes to them and says, hey, we want to know the number of deer in this area, they can say, hey, we've got the most efficient method used in counting deer that's going to save you cost, which is, if you're in an industry where you have to bid for your work, then you have to have a way to keep your cost lower, but still a what's my word there? A desirable outcome. You know, you got to have a good product. You can't just be the cheapest of something, but you still got to be a great product at the best price. So that's the purpose of all of this thing. So the question, based on the research that they did with other counting methods, the most prevalent counting method was a 6 to 11 night rotation basis of counting deer. Up until this time, the thought process was we need to do it a bunch of times in order to make sure that we get a good mean, a good average, a good set of data to do so. But to take a crew of 5, 6, 8 people plus the equipment out that many nights, it's a pretty expensive investment. And so these two guys, Raymond and Richard, were looking at, would a shorter period of time produce similar results to those longer periods of times. And you can see here in the question that I have right here, this is the question that they asked in their journal article. Would spotlight counting in small periods produce similar results as counts in longer periods? Therefore, in other words, if we can do it in a shorter amount of time and get the same results, then we're going to have a lower expenses, and therefore, when we produce a price for a company, it's going to be lower than those that are out there trying to do it 6 to 11 times. So what that really points to us and remind us that there are a lot of things that are determined scientifically, but they have commercial ramifications. And this is the case here where these are scientists, yes. These are biologists, yes. But what they're trying to do is have a good business policy, a good business model, so their company, their organization can be successful financially. Even though it may be a nonprofit, okay, they still need to cover, they still need to produce revenue so they can have salaries and equipment and operate in a day-to-day manner. So this would be no different than a plumber trying to decide if you think back to plumbing, and if you're only 20, this you can't even fathom this. But at one point in time, all plumbing was done with metal pipes. Okay, and then someone figured out, hey, plastic pipes, PVC pipes, are a whole lot less expensive, and they can do just as good a job, and they can last just as long as metal pipes. And plus, they won't have long-term ramifications as far as health and consequences into the water as the metal starts to rust and all that kind of other stuff. So someone figured that out. So now you've got, at some point in time, you had plumbers who were still using metal pipes, some plumbers who had turned over to the plastic pipes. And if people building homes were getting bids from both types of companies, the plastic pipe, the PVC pipe plumber was going to have a lot lower bid than the metal pipe. And so it kind of just, that's the same thing that's happening here. That was a long way of saying that, but hopefully that gives you a good sense of the background of what's going on here. So, hypothesis, okay. Their hypothesis, if we spotlight count for 2 to 4 nights, then we're going to produce similar results to counts of 6 to 11 nights. And when we talk about, and you can kind of see those on the right-hand side, when we talk about what a hypothesis is, we're going to compare it to what they have here. So going back to our article, because some other things that you want to spotlight, or no pun intended there, is something like this right here. Okay, this is a short indicator as the importance that math is going to play, because they're going to do all this research, and they do research, and you can see it right here. 17 years worth. Well, they say 18. Oh yeah, because this would be year 1. This would be year 18. Yeah. So 18 years worth of data. Can you imagine doing this for 18 years? Some of you that are listening to this aren't even 18. So can you imagine your entire life going out, counting these deer, recording the data, and doing all that stuff? But anyway, but the idea is these two guys, they're not going to collect the data and just decide in their head, hey, yes, it did work. No, it didn't work. Mathematics is going to be what determines the outcome here. And then something else, other things to emphasize. Not only were they looking to decide yes or no, does this work, but there was a lot of other things that they also looked at as far as influencing their numbers. And we're talking this environmental parameters, temperature, humidity, wind speed. You're going to see that they looked at acorn production, since that's the main food source. They just looked at a lot of different variables, just trying to really understand what the numbers were telling them. All right, so let me close my ink tray here, and let me slide this over so we can see these two parts. Now, that there are 6 of these little journal article screenshots, and that was the first one. So if you want to gauge on how I'm doing on time, that might be a good indication. But the end, the last three are a lot shorter as far as time than the first three. So it probably isn't a great overall indicator. But an observation, I've already kind of touched on this, something that you notice that raises a question. And of course, it is obviously done by the five senses. We don't, anything that we recognize is within the five senses. I mean, you just take sight and hearing alone is a majority of all things that you sense. But then you throw in touch, that would be the third one, and then of course smell and taste are we recognize things in our environment a lot less than smell or taste. Now, if you were a dog or something like that, snakes, for example, that might be reversed a little bit because they rely a lot more on their smell and taste to recognize that than we do. We don't have to taste something first to understand what it is, or smell something first. And if you've got a dog, you certainly understand what we mean by that. Then when it comes to a question, okay, the key thing about the question part of the method process is that it's got to be something that can be measured. Okay, a scientific experiment is not going to evaluate coolness of something or the popularity of something. You can determine what's a student's favorite type of pizza is by giving them options and seeing what they choose. That you can measure. But what you can't measure is that does pepperoni taste good? Okay, is pepperoni popular? Is Taco Bell cool? That kind of stuff. So it's got to be something that can be measured. And then when we talk about a hypothesis, it's got to be an if-then statement. Okay, it needs to be a prediction. It's got to be testable, and eventually it's going to be compared to the data. And if you look over here, we got our if-then. Okay, we make a prediction, and then we're going to produce similar results. Okay, and we can test this, and you'll see all that stuff. So, but again, if you're a high school student currently or a recent graduate, these kind of things between middle school, high school, probably 2, 3, 4, 5 times you went through this process. So let me slide down here to this next section here. Quick drink here. All right, so lots of highlighted stuff here. But we've kind of now set up all the background information and we've kind of set everything in place. And now we're to our experiment. Now, it would be helpful in journal articles if they use the word experiment, but they don't do that. Whenever you pick up a journal article, when you look at the ones we're going to look at, the word methods is that section heading, and it's more about the journal article wants to talk about what we did. Okay, not here's our experiment. But anyway, because it's more than just the actual steps of an experiment, it's more about all the things that they're looking at. But anyway, so the word method, let me get me a pen here, refers to the experiment. All right, so some things that they did, they're going to set up here in this first yellow highlighted section, what they were going to do. Okay, conducting fall counts from 81 to 98, time of the year that they were going to do this, and then they get into the equipment or the when, 2 days, 8 surveys a year, the type of equipment, the spotlights, what's powering the spotlights, and then they also get into more equipment over here because they're also, as we mentioned, they're going to record temperature, humidity, and somewhere down here is wind speed. Wind speed right here. So they've got, not only they got these very powerful spotlight flashlights, they've also got equipment on the truck that's going to measure temperature, humidity, and wind speed, and not just ever so often. These are scientific equipment that's recording it constantly. You can see here every 2 seconds, um, every 2 seconds. So that's pretty significant data collection going on out there by pretty intense equipment. So not just somebody looking at their watch. Oh, the phone says it's 78 degrees, so write that down. No, it's being collected scientifically in a very efficient, powerful manner. Okay, it also down here talks about the crew and what they've got going on. So this leads us into conversations about variables. Now, the three main variables to always recognize and talk about is the independent, the dependent, and then the control variables. Okay, so the independent variable, that's this independent, dependent. It's always difficult for students. I should say all, but for some students to get a grasp on what we're talking about when we say independent, dependent. And the easiest way for me to remind students about it is one of them is the data that you're going to collect. The other is the thing that you were testing, okay, or the idea that you were testing. So idea for independent, data for dependent. Okay, so dependent is easy. That's the numbers, and I've got that referred to right here. That's the numbers that you're going to collect. So in this case, as you can see here, the dependent variable is going to be the number of deer counted in each of the sessions. Okay, D for dependent, D for data. Okay, straightforward, pure and simple. Independent, okay, we're going to think about I, and for idea, the idea that you're testing, and writing with the mouse cursor, it's a great thing. So if you think about this experiment, what are they testing? Okay, they're testing to see if the 2 to 4 nights is just as efficient or just as accurate as the 6 to 11 nights. So in this case, the independent variable, okay, you could say simply the number of nights counting. That's probably more accurate, but in this case, the independent variable is the 2 to 4 nights testing, because they don't actually go out and count for 6 to 11. They compare that, and we'll show you that. They compare that to previous data. And actually, I shouldn't say previous. The 6 to 11 data is constantly occurring at the same spot during the same time frame. So the beauty of this journal article, they're able to compare apples to apples. Okay, not apples to oranges or apples to something else. Another company is conducting 6 to 11 research numbers right along with the 2 to 4. And there are some times that they're out there at the same time. So they can really compare numbers based on it being such a similar situation. So then for the control variables, and I'm right here, the control variables, it's super, super, super important to have your variables controlled as much as possible. Okay. So for example, if in this case, the control variables, and I have them listed here, is the equipment used. Okay. And that's super important. The beginning time that they go out each night to start, how long they're out there each night. Okay, it's important that all that is controlled. It's done the same way every time. That way, when there's a variation in the data, you know it's for something else and not because of you didn't control the variables. So, for example, when they say that they begin 1 hour after sunset and they go out for 2.5 hours, if this when they begin was random, okay, and then the numbers were different, they wouldn't know, is it because population numbers are changing, or is it because we're going out at different times, we're seeing different results from everything, or the length of time. If they would go out sometimes for 4 hours, sometimes for an hour, sometimes for 3 hours. Again, the data difference, you would not know truly what was causing that. Another quick drink there. Oh, I didn't even think about that. We could monitor my time based on the clock down here. Or some of you have probably picked up on that. I didn't think about that. So that's the idea behind the variables. Now, one thing that if you were just to look this over, and we'll talk more about this in class, is that one thing that would have been better, it just jumps out, is that it should have probably been controlled, is this right here. So if I tell you, and I just identified it, I just highlighted it for you, if it's known information that some nights they have more crew spotting deer and other nights less crew, and if you've got one person counting on one night and then you've got 4 counting on the other night, what's the obvious thing that should jump out at you? And I'll pause here while you think about that. But the obvious thing is that, hey, if I got 4 people, there's a chance that we see deer that the one wouldn't have seen. So that would probably have been something that would have been something maybe to control. But maybe that wasn't a possibility. People can get sick, or they've got other things, or just something would come up, or who knows, maybe they're all out there and something happens. Somebody gets injured or gets called away, emergency, whatever. So that may not have been something that could have been controlled. And based on their conclusion, their findings, it didn't affect the overall situation. So let me close my pen here, and we will scroll up and over to our next. No, I didn't want to do that. To our next scenario. Yeah, if you, in trying to move over, if you put your cursor on something that's there, it will think, oh, I want to move it, as opposed to just moving over. So we've had our observation, we've had our question, we did our hypothesis, we did our experiment, okay, and now we're going to begin that process of analyzing, okay, what we're doing. And you can see here, and this is just the beginning of it. It's also, if I scroll up here, you can see that we continue down there. Okay, we're going to begin to compare the data, okay, and again, emphasizing mathematical. It's not so obvious here, but it is down below when we look at this. But some cool aspects, and I probably used the word cool wrong, but anyway, that's how it goes. But some interesting aspects of this is, I did it again. Oh, I don't have my pen up. That's why I didn't respond that one. All right. So in this section here, it talks about other ways that, or some of the many ways that people counted. You can see here some use pellet groups, trail counts, drive counts. Excuse me. Some relied on just counting tracks of deer, aerial counts, ground-based photography, hunter observation, daytime vehicle route counts, nighttime counts. There are just a lot of ways that people were using to record the number of deer. And then right here in this section, this is what the whole thing is about. The diversity of methods is the result of researchers seeking accurate, easy applied methods that are cost effective, implemented across differing landscapes. In other words, people doing all these things, trying to find the best way. Okay, the best way for the least amount of money. Okay, that's this whole gradient is going on here. And they even mentioned the purpose of this whole thing. These 18 years is just simply this: to determine the trend in deer abundance in heavily managed areas, to generate precise representative counts, okay, for the money. And they also kind of focused on the abiotic, biotic stuff, what's going on, things that are living, things that are not living, how they affected the deer populations. So super, super complex, interesting stuff. Okay, now we get into the meat, okay, of the analysis. And you see I almost got this entire thing highlighted, and we'll just kind of walk through it. It's not meant to be an in-depth memorization, but and you can already see, I don't have my pins up, in sections like this, and you can see here and over here, heavy numbers. Okay, heavy numbers. And that's because when we do analyze, it's all about the math. Okay, it is completely about the math. No opinions, no gut feelings, no personal choices. Only math determines the conclusions. And if you don't do that, and you try to produce a scientific paper based on your opinions, it will not see the light of day. When it goes through the peer review process, other scientists will say, no, you didn't use any science here. You go back and try it again. We don't want your opinion on the best way to count deer. We want scientifically proven, data-driven processes. You don't have that, you don't get to be in the fancy magazines. So let's look at some of the things that they did determine in the way that they talk about it. So we start up here at the top. Count of deer for all trips were averaged to produce an annual mean. So again, math. To examine the stability of our count within years, coefficient of variation. In other words, they not only looked at total numbers and averages, but also how much of those numbers differentiate, and then tried to look at why they differentiated. Was it food resources? Was it heavy hunting years, the previous years? All those things that kind of play into what's going on. And then they sprinkle in here some findings. And one of those is right in here where they test the hypothesis that if you have increased corn production or acorn production, then you're going to have increases in your observations, or they should decrease because the deer are driven to the tree areas, the forest, and you're not easy to be seen as opposed to the open fields. But again, the take-home measure here is that it's math. It's all about the math. You don't have the math to back up your findings, then your findings aren't considered to be legitimate. So that's what we want to drive home. Now, I mentioned this earlier, but this whole thing right here and this section of highlight is the explanation of what this graph is. This graph here, at first thought, you'd think, okay, that's their graph. No, this graph is for the data that Mary's group did, but what they were doing is the 6 to 11 night rotations, and you can see that the years are exactly the same. So when these two guys comparing data, they're comparing data from the same area, the same deer, okay, just a different amount of time being spent on it. So they can really compare with the belief that they're comparing true data, because it's the same areas, same deer, same processes. The only one changes the time spent out there. So in other words, all the variables are controlled except for one. That's the independent variable. That's the one you're testing. That's the idea that you're testing. All right, so we don't want to get heavy into the math, but just recognizing that all these things are math driven. Okay, all these different variables that they're looking at, it's all about the math that decides yes or no. Okay, so let me close my deal and go up to our last two. And again, as I mentioned, and you can see here, there's not even any writing out there because we're just going to pick up on a couple of things. But I just wanted to kind of highlight the method used to determine conclusions based on this scientific process, and then using the journal article to kind of show you what it looks like. And again, we'll do this again in the classroom. But again, heavy math right here. Oh, I don't have my pens opened up. Heavy math right here. But we're really going to start to kind of highlight our findings or identify our findings. High correlation coefficients exist regardless of whether the trips were in 1 week. But anyway, so we start right here. Okay. Found that the deer were increasing 2% per year. Now that's significant because when I talk about comparing true data, if I go back over here, it's down here. You have to this graph here, and you can't see it. Well, let me just go bigger. Yeah, so you can see it. And again, I keep forgetting to open my pens up, but it's right here. Okay, 0.019. I probably can't really see that, but if that's a percentage, okay, or can be converted to a percentage. You got this slope here, and you can correlate that to the 2% increase. So close my pen here. I'm really breaking down on my remembering what I got to do here. No, we need to be up here. So when I say comparing data, they were really able to compare and see true results. Something else that, and we'll highlight it down below here as well, that we want to highlight. Not sure why that did that. Oh, because I was trying to write without my pens being up. Anyway, they referenced a lot of journal articles in these, and these previous journal articles were used to set up their experiment, talk or determine their equipment, things that they'd want to evaluate, what they would record. So in other words, these guys just didn't think of, okay, this is what we want to do. They used previous articles in scientific journals to determine what they wanted to find out. And that's the way these things work. You look at all that's been done previously, and then you use that to make sure you're designing a good quality experiment. And in the future, somebody will use this article or has used this article to make their experiment more significant. But let's see. And then some other things we talked about, some other things in the past that people have recorded as ways that they had errors in their data, detection rates and things. These articles talk about. So these guys put those findings into place to try to reduce any error that they might have in their data. Okay, and over here, all these articles, these previous articles, refer to other factors that can influence or affect spotlight count. And so again, they use those findings to make sure that their experiment procedures, their setup, was the best that it could be to try to eliminate any errors or mistakes or overlooking things. Okay, and then they put in a little finding here that they confirmed that if there are years where there's heavy acorn production, that the deer leave the open fields trying to find food and just go where the major food source are. But they also talk about that that's something that needs to be studied even further, more directly, instead of just being side angles into experiments. It needs to be something that is looked at more efficiently. All right, and one more here, and here we go. All right, so, and I don't know if you can hear the dog barking in the background. That's Avery, our 90-pound lab, who, if anything, if she can hear it outside, speaking of the five senses, if she can hear it or see it outside, she's got to bark at it. And trust me, when the mailman shows up, we all know it because she goes bonkers. But anyway, that's a side topic. You didn't need to know all that. But this section right here, these two yellow highlighted sections here, are going to show you just how important these processes are. And you may think, wow, that's a lot of work. But all this work, think about the time, 18 years, a 10-page journal article, all the readings, and this is our cited section. And I just have the first part here, A through D. But there were two more pages of all these articles that we had to incorporate and read and use to build our experiment. So we're talking a massive amount of effort and time and energy went into all this, and all that work went down for these two sections right here. Okay, means of the initial 2 or 4 trips were similar to means for all trips 6 to 11 days. So that confirmed their hypothesis. Okay, their conclusions was positive, but just think of all that time and energy, okay, went just to produce that sentence. But because of all that they did, their findings are highly esteemed and held because they put such effort and energy into everything. See, such similarities in counts indicates wide flexibility for managers in applying this technique under similar conditions. In other words, they're telling you that, hey, we have found the most cost-efficient way, but it's a way that produces similar results to the other ways, to the old ways. So we can get you good quality data, but we can do it in a much more inexpensive manner. Our data also revealed that spotlight data from as few as 2 to 4 nights can still provide reliable information for managers. This reduced effort can save agencies both time and money when using spotlighting for indexing deer abundance. And surely, or I shouldn't say surely, but I'm sure you can understand that if you were trying to talk a company into buying your product, the fact that we could save you in this situation both time and money, yeah, they're going to go with that. So, but anyway, a lot there, well over an hour. But the idea is to kind of understand the scientific process that is used to determine results, but not only just giving you the details about it, but showing you how it looks in a real life scenario. That was the purpose of all this. And like I said a couple of times, probably way too many, but once we get together in classroom, we'll have this one, plus we'll have others that we will look at and just evaluate the question, the observation, the hypothesis, the analysis, the conclusion, and the experiment, all that stuff. We'll look at it to just kind of get a good sense of what it looks like in a real life scenario. Well, there you go. Chapter 1 is in the books as far as a lecture. The next time we come together in this format, we'll be looking at Chapter 2. And the beauty of Chapter 2 for me is I can use it with you guys, but I can also use it with my A&P students because they also have that same chapter in their textbook. But it's all about the chemistry. And you may already be thinking, dang, we just had math, had a bunch of math talk. Now we're going to have chemistry. And you may also be thinking, whoa, 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 whoa. Why are we talking chemistry in a biology classroom? And that is the question of the ages, because that answer is almost the answer to life, is that everything that happens within our body, our cells, everything from our brain sending out messages to the breakdown of our food molecules to the determination of do I need to, or even T cells deciding who should we attack or not attack. It's all done through chemical reactions. And man, it is fun to talk about. I love this chapter 1. I like talking about this stuff. But chapter 2 is such a great time to talk about. Chapter 2, 6 through 8, when we talk about energy, man, I just love chapters like that. But anyway, we'll get to all that. So next time we talk to each other, it will be about chemistry. So just a little, I don't even know what word to use there. If you're a high school student who has had chemistry, you had it last year, or you're having it now, you've had it in the past, or you're a recent high school graduate and you had chemistry, a lot of it will seem pretty similar to you. What we're going to talk about in the chemistry setting is probably just basically like the first month of your chemistry class. Nothing is thoroughly deep, just the important aspects of what happens in a cellular structure from a chemistry standpoint. Well, but with that, let's shut this down. Let's get you out of here and back on the road, doing whatever it is that you're doing. Anyway, again, thank you. I hope you enjoyed that. I enjoyed it. Look forward to seeing you in the classroom. Oh man, you don't know how much I'm looking forward to seeing you getting back in a classroom, a good school structure. It has been so long since and like in the high school setting, we went to spring break and never came back. Oh man. And Crowder, we just kind of shut that down. We weren't in. No, Crowder's in spring break as well. Yeah, and we just never came back. So, yeah, but it's a serious situation. Can't make light of that, and it's a potentially dangerous situation for a lot of people. And even now, it's August 1st, as you can see here, and just even locally. I mean, the Cardinals game getting shut down yesterday. Still little hot pockets all over the state, all over the country. Plus, right now, Florida going through a hurricane, so sometimes you just got to do what you got to do with what's going on. But anyway, look forward to seeing you in the classroom. And with that, I will finally say goodbye.